Hello everyone, welcome to Botany option channel for UPSC examination. In today's video, we are going to focus on the seed germination. As you know, this is an important part in the syllabus of various examinations. So you should prepare this topic very carefully. Before going into the seed germination, I request you to join the Telegram channel of the same name, that is the Botany optional for UPSC examination. There, we continuously upload the links of these videos plus the PDF files relating with the botany channel so that you will not miss any study material relating with the botany subject. All right. So let's learn about the seed germination. Now, first of all, we are going to see what is seed germination. Now, seed germination is an process in which the dormant embryo of seed reassumes its metabolic activity and grow to produce the seedlings. That is, the seed germination is nothing but the process in which the dormant embryo of seed reassumes its metabolic activities to grow and convert into the seedlings. This does not happen so simply because the dormant seed, the all the metabolic processes are arrested, right? No metabolic processes is occurring in the dormant seed. Now, what triggers these activities? The very first steps in the germination of seed is the water imbibition by the seed. That is, water is imbibed by the seeds, and due to the imbibition of the water, the metabolic activities of these dormant seeds starts to produce the various biochemical products. Now, what happens due to this water? All the hydrolytic enzymes which are present inside the seed, they gets activated, and due to the activation of the hydrolytic enzymes, these hydrolytic enzymes converts the complex chemical compounds which are present in the seed into the simple compounds such as glucose and maltose. This food material, which is now available in the simple form, that is in the form of glucose and maltose, is utilized by the seed because photosynthetically seed is yet not active, and that is why they cannot prepare their own food. Now, imagine the condition. Here is the seed and it does not have any leaves, right? As you know, leaves are the primary sites in the plant kingdom where the photosynthesis occurs. Now, the bare seed, this seed does not have any kind of leaves. That is why it cannot perform photosynthesis. And due to the no photosynthesis, there will be no chemical compound formation. But this seed imbibes the water, the hydrolytic enzymes which are present in these seeds got activated. And what these hydrolytic enzymes do is that they convert the complex chemical compound which are present in this seed into the simple organic compounds such as the glucose and maltose. Now, these glucose and maltose are utilized by these seedlings during the non-photosynthetic phase. Now, due to the water imbibition and use of these glucose and maltose like chemical compounds, the growth in the seed takes place. Due to the growth, there is a huge pressure on the seed coat. Now, due to this huge pressure, this seed coat ruptures. Now, when this seed coat ruptures, the radicle comes out of the micropyle and develops into the root system. That is, this radicle finally converts into the root system, and the plumule which comes out above the soil surface is converted into the stem, that is the shoot. So the plumule converts into the shoot while the radicle converts into the root system. Now let's consider again how these things happen. First of all, we have the dormant seed, and there is no metabolic activity in these dormant seeds. There are many complex chemical compounds plus the hydrolytic enzymes are present in these dormant seeds. But when these seeds imbibe the water, these hydrolytic enzymes got activated. Now, what happens due to the activation of hydrolytic enzymes? These hydrolytic enzymes converts the complex chemical compound into the simple chemical compounds such as glucose and maltose. Now, due to the utilization of these simple chemical compounds plus water, the seed coats gets ruptured and the radicle and plumule comes out of it. Then the radicle got converted into the root system while the plumule is converted into the shoot system. So this is how a dormant seed finally converted into the seedlings. All right. Let's consider the types of seed germination. Now there are main two types of seed germination in which the seed germinates differently. The first is the hypogeal germination and second is the epigeal germination. Now the main difference between the two is that whether the cotyledon remains under the soil or whether the cotyledon comes out of the soil. Depending upon these, these are two types are there. That is, first is hypogeal germination and second is the epigeal germination. We will see these two types one by one. All right. Now let us first consider the hypogeal germination. Now in this germination, the epicotyl elongates quickly and pulling the plumule out of the soil surface while the cotyledon remains inside the soil. Now as you can observe in this first picture, this seed starts to grow. Now after the growth, what happens? The epicotyl elongates. As you can see in this picture, the epicotyl is elongating. Now due to the elongation of epicotyl, what happens? It pulls the plumule out of the soil. That is, it pulls this plumule out of the soil and it goes into the air. While doing this, the cotyledon simply remains under the ground. And then this plumule which comes out of the soil surface got converted into the whole shoot system. At the very same time, below the ground, the radicle comes out of the seed and then this radicle slowly converted into the root system. So we have here the shoot system above and root system growing below. So the very important point in the hypogeal germination you have to remember is that the cotyledon always remains below the ground while the plumule goes to form the shoot system while the radicle forms the root system. Actually, this word hypogeal literally means, hypo means below, while geal means soil. So the which remains below the soil. That is why this type of germination is called as the hypogeal germination. This hypogeal germination can be observed in monocotyledons as well as the dicotyledons. In both the groups, this type of germination is present. In the monocot, we have the examples of rice, wheat, etc. While in a dicot, we have the example of pea, mango, etc. So the hypogeal germination occurs both in monocotyledon as well as in the dicotyledon. The example for the monocotyledon is the seed and wheat, while the example for the dicotyledon is a pea and mango. All right. Now let us consider the second type of seed germination, that is the epigeal germination. Now in this epigeal germination, the hypocotyl elongates. This hypocotyl pulls the cotyledon and plumule out of the soil surface. That is, these hypocotyl not only pull plumule, but it also pulls the cotyledon out of the soil surface. As you can observe in this picture, here is the seed which is germinating, and after the hypocotyl formation, the hypocotyl pulls not only the plumule but the whole seed out of the soil, and that is why this seed goes into the air, that is in the shoot system. It does not remain inside the soil. So in the epigeal germination, the cotyledons comes out of the soil surface. Epigeal word is again meaning, epi means above, while the geal means soil, the one which is above the soil. So the cotyledon in the epigeal germination goes above the soil. And again, this is a very basic difference between the hypogeal and epigeal germination, that in hypogeal, the cotyledons remain below the soil, while in epigeal, the cotyledon goes above the soil. All right. Now, when these cotyledons goes out of the soils, they become the flat and they perform the function of embryonic leaves. That is, these cotyledons once comes into the air, they perform the function of embryonic leaves and they start to prepare the photosynthetic food till there is a formation of new foliage. That is the original foliage leaves. Till their formation, the embryonic leaves, which are nothing but the flattened cotyledons, perform the function of photosynthesis to produce the food. All right. Now, this type of germination can be found in many dicot plants, and the example for this is a castor and tamarind. All right. So you should remember carefully, in the epigeal, the cotyledon goes above the soil surface and converted into the flat embryonic leaves. These flat embryonic leaves make the food till there is a formation of main foliage leaves, which can literally perform the photosynthesis. All right. Now let's compare the hypogeal and the epigeal germination for the more understanding about these are two types of germination. This area showing the epigeal germination, while this picture showing the hypogeal germination. As you can observe, in the epigeal germination, the cotyledons comes out of the soil, while in a hypogeal, the cotyledons always remains below the ground. This is the very first difference. In the epigeal, the hypocotyl plays an important role of pulling out the plumule and the cotyledon out of the soil, while in the hypogeal, the epicotyl alone pulls the plumule out of the soil surface. So this is the second difference. So both this type of the germination, which can be found in a monocot as well as the dicot, shows two basic differences. First is the cotyledon, which may remain inside the soil or above the soil, and another is the in epigeal, the hypocotyl plays an main role, while in a hypogeal, epicotyl plays the main role. Now let us see the very special type of seed germination which is often occur in mangrove plants. As you know, the mangrove plants are the very special kinds of plants. These remains always along the coastal lines of the salty water. That is, they found along the coastlines of the oceans. And these mangroves shows the viviparous germination. This is very interesting because this is not relating with the epigeal or hypogeal germination. It is completely independent, another type of germination. What happens in a mangrove plants when the fruits are born on the tree? All these are the fruits which are brown in color. All these are the fruits which are born on these trees. Now, the seed starts to germinate when the fruit is still on the parent plant. That is, the fruit is still present on the parent plant, but from this fruit, the seed starts to germinate. You can observe in this picture. See, this is an mangrove's fruit, and this fruit inside contains the seed. Now, if you observe carefully, this fruit is still attached with the parent plant. This seed, which is present inside the fruit, it starts to germinate. Here you can see the green part is coming out of the seed. The seed which is present inside the fruit, and that fruit which is still connected to the parent plant. In mangroves, these seeds starts to germinate. So when they start to germinate, they form these green areas, and the radicle comes out of the seed, and they form this elongated structure. So all these are the new mangrove plants which are still on their parent plants. Now, why this happens? Now, what is the reason that such a type of seed germination is occurring in the mangrove plants? Now, there is a reason for this. This is the evolutionary adaptations in the mangrove plants. As you know, mangrove plants are always growing along the coastal lines, right? And below there is a floating water, floating salty water, and this salty water is not suitable for the seed to germinate directly into this salty water. So what mangrove plant did is that they germinate their seeds when they are still on the parent plants, and once they forms the particular shape like this, then they fall off this structure into the water. Now what happens due to this shape? As it falls into the water, it grows directly into the soil. Now due to the attachment to the soil, this radicle starts to grow quickly and have grip on the soil, so that it will not float away by the water. And once it has the grasp on the soil, it then grows into the new mangrove tree. So this is how the mangrove plants avoid the loss of seeds by germinating their own seeds while they are still collected on their parent plant. So this type of germination is called as the viviparous germination, which is commonly found in mangrove plant. All right. Thank you very much for watching this video. Please like the video. Please share this video with your friends who are studying botany. And if you haven't subscribed to our channel yet, please subscribe to the Botany optional channel for UPSC examination. Again, thank you very much for watching this video. See you in the next one.